Ever twisting, turning, and at war with itself and external invaders, DNA provides the fuel for evolution's changes. 10% of our genome made up of ancient viruses, and at least another 60% consist of repeated elements made by jumping genes gone wild. Only 2% is made up of our own genes. But within cells and genetic material of different species, merging and genes continually duplicating and repurposing, life's history flows. And as an evolutionary biologist put it, more like a braided and meandering river rather than a straight channel. So, paying tribute to this journey, the Calcutta Consortium on Human Genetics and Birla Industrial and Technological Museum, a unit of National Council of Science Museums, Ministry of Culture, Government of India, warmly welcomes you to the interactive session, DNA Celebrating Our Shared Humanity. We are privileged to have with us a stalwart of the field. Let us welcome, with a big round of applause, Professor Partho Protim Majumdar, National Science Chair, Scientific Excellence, Science and Engineering Board, Government of India, and founder, National Institute of Biomedical Genomics, Kalyani. We look forward to learning from you, sir. Time to introduce the hosts of the session. Please welcome with a round of applause, Professor Arindam Moitra, President, Calcutta Consortium of Human Genetics, and Professor Bhashwati Pandit, Secretary, Calcutta Consortium on Human Genetics, Shri Arnab Chatterjee, Director, Birla Industrial and Technological Museum. Before we engage in this interactive session, I welcome to the podium Professor Arindam Moitra, President, Calcutta Consortium on Human Genetics, to deliver his welcome address. Over to you, sir.
Thank you so much. Without further ado and without wasting much time to the fantastic feast that lies ahead, let me welcome all the dignitaries and teachers on and off the dais and the collection of young minds we are all fortunate to have you all today here. The Calcutta Consortium of Human Genetics is a very well-known group that started its journey somewhere around 1999. Parthuda and Kunalda others can correct me if I am wrong. I was also just freshly out of PhD then, but not in Kolkata. And this came up as an offshoot of the research that were being initiated for the first of its kind and first time in India by people, some of whom are present today. We are fortunate to have with us today, primarily from the institutes of Indian Statistical Institute, Indian Institute of Chemical Biology, Science Institute of Nuclear Physics. So they all wanted to study the human genome variation in the context of populations and in context of human health and disease across India. In many ways, this city that gave birth to this kind of studies, and this consortium was the first of its kind and showed the path that human genomics and human genetics in India would not take subsequently. Because following on the footsteps of these kind of initiatives, the first major human genetic initiative at a pan-India level was started by CSIR, what is known as the Human Genome Variation Consortium, also nicknamed that time as the SNP Consortium. Again, these stalwarts played a leading role in that. And then subsequently today, you have all heard in the newspapers about the 10,000 Indian genomes that have been catalogued and sequenced. And again, one of the stalwarts of that is present here in the hall. So, Anulava, can you please rise up so that people know you? You tend to hide. Yes. So this whole bunch of us are actually all engaged in some way to understand the human genome, how that dictates each of our health and disease, and the diversity, the fantastic diversity we witness. Even in this room, if you witness the diversity, and that all emanates from the human genome. And that is what we are very passionate about, and that is what we want to show you, or we want to share with you some of the things that the human genome is all about. And I'm pretty sure that in the classrooms, these things are not taught, and today you will remember for many time to come. Without any other thoughts, let me thank our director, BITM, and other members of his team who put up a fantastic job. And it is because of them that we could hold this today, and we look forward to many more such events in the near future. Welcome, Nitaida. Thank you very much.
Thank you, sir. Today's session will not have been possible without the impetus and caring guidance of Director BITM. May we now request Sri Arnab Chatterjee to please address the audience gathered for the session. Over to you, sir.
Thank you, Aditi. Respected PPM sir on the dais, distinguished scientists on the dais and off the dais, my dear students, teachers, and colleagues. I'm privileged to be a part of this event here today. And to be very honest, my knowledge in the subject matter is very shallow. So I won't prefer to, in front of the stalwarts present here, I won't prefer to put up a word on this subject matter. Rather, what I mean to say is it's an enormous opportunity for all the students present here, because in our childhood or as a part of our curriculum, we get to learn a lot. We get to study a lot about from our textbooks. But what is written out there in the textbooks, we fail to understand many a times what kind of work went behind that. And today, here it is your opportunity to listen to a person who are scientists and who are there as the forerunners of activities of our country. So instead of just the science entirely is not about mugging up a few facts and then reproducing them on the papers and all these things. It's about once you get to live the life of a scientist who was instrumental or who were instrumental behind making this happen, behind the formulation of such theories and other things. You won't Once you get to learn about the theories, you have to live the life of that scientist. I mean, then only your knowledge of sphere is complete. So here it's a one-time opportunity. It's, I mean, kind of an opportunity for you all to listen to the stalwart scientist in this field of genomics. And the second thing I'd like to mention that in due course of your career, you may be opting some other subjects other than science. And the only thing you should not stop doing, I mean, it's the magic word, the three-lettered magic word, that is why. If you start asking why, you'll get to know many things which is beyond your boundaries. And you all know that a well-framed question is much better than 1,000 silly answers. So I'd like to request all of you to pay your attention to this particular talk and ask whatever comes into your mind. Even if it won't be possible for us to cater to all your questions, maybe we will. You can drop us an email. We'll respond to that, and we'll try to. Cater to you the best way possible. Without further ado, I'd like to stop here because I won't like to stand in between you and the speaker for long. And thank you. I hope you're going to have an enriching session today. Thank you.
Thank you, sir. To delve into the nuances of genomics and learn what this excitement is all about, we have with us a master of the field. May I now request Professor Bharshati Pandit, Secretary, Calcutta Consortium on Human Genetics, to introduce today's speaker to our audience.
Good morning, everyone. Today we'll listen the story and some interesting facts about genomics, and Professor Parthupratim Majumdar is going to give us the essence of that beauty and the um, beauty of genomics to you. So let me introduce to you, as many of you may not have heard much about him. So Professor Parthupratim Majumdar is an internationally renowned human geneticist. Currently, he is a distinguished professor at John C. Martin Center for Liver Research and Innovations in West Bengal, and also the National Science Chair, Government of India, and member of technical advisory groups on genomics, World Health Organization. He has made seminal contribution to human, statistical, and population genetics, genomics, and led several national and international research projects. He founded and directed the Research Institute, National Institute of Biomedical Genomics in Kollani, which is solely devoted in research in the area of genomics. He is also one of the mastermind and leader for the formation of the Calcutta Consortium of Human Genetics, as you heard a few minutes back. He is also associated with several academic institutions like Indian Statistical Institutes, ISERS, Jawaharlal Nehru Center for Advanced Scientific Research, and also served as a president of Indian Academy of Sciences. He is also bestowed with several awards, accolades, and membership of many national and international prestigious scientific societies. We are honored to have him amongst us, and we'll hear from him, genomics, what's all this excitement? Thank you, and I hope you are eagerly waiting for the talk.
Thank you, madam. We'll take just a little time to clear the settings. I'll request the dignitaries to please be seated in the audience while we set up the stage for the speaker.
Yeah, good morning, everyone. The way that we have conceived of this program is that we will show you a film, a very short film, on the history of the Human Genome Project, when the first human genome sequence or sequencing of the first genome was conceived. It was preceded by a large number of sets of discussions, some positive, some negative, etc. Eventually, it was undertaken by multiple countries, and the history of that particular sequencing project is narrated by some of the leaders of the Human Genome Project. So we'll take a look at that, followed by I will give a talk on the excitement of genomics today, and that will be followed by some 10 bold predictions that have been made by Dr. Siddharth Mukherjee, whose name many of you may have heard. If not, I will, or somebody will introduce him to you before we show that film. So this is what the program is all about. And let me take this opportunity before I bring the film onto the screen. Let me take this opportunity to thank the director, Sri Arnab Chatterjee of the Birla Industrial and Technological Museum, and my dear friend Aditi, who actually organized all of this. She belongs to this, and she's in the forefront of getting science to the public, especially among the school children. So public outreach of science is very important. Those of us who belong to different laboratories. We do our work in these laboratories, but very rarely do we get out of our laboratories to explain to other people what we are doing in the laboratories. And now many of us believe that that's absolutely essential for people to know what we are doing, and not just that, to enthuse school students to belong to our community, to do science. Not just the kind of science that we do, maybe other types of science, but if you know one kind of science, how that's cultivated, you would actually become interested in other kinds of science that may interest you as you get exposed to these variety of sciences in your schools and colleges. So that's essentially the purpose, and Dr. Aditi Ghosh plays a major role in the public outreach of science, and she's helped us organize this in addition to the Calcutta Consortium of Human Genetics. We have heard the president of this consortium right now, Dr. Arindam Moitro, the secretary of the consortium, Bhashwati Pandit, and others. And this is a whole group of us who actually try to move this human genetics forward through these public outreach programs. We are very glad that you're here. And so without further ado, let me start the film. But one more announcement before I start the film. Many of you may already have seen the notice. Some of you may have seen the actual exhibition. There is a fantastic exhibition that's going on, that's been put together by the National Council for Science Museums, which is funded by the Government of India, promoted by the Government of India. There is a fantastic exhibition that's going on the history of vaccines, vaccinology, and how vaccines have helped public health. Please, this started about a week ago, and it will be there on exhibition in Science City for 6 months. This has already toured 4 different Indian cities, and it's right now at its fag end in Science City. And after this, the entire exhibition will be dismantled, unfortunately. So this is your turn to go and see what vaccines do to our lives and how vaccines are made, starting from the history of vaccinology to the most modern predictions of where vaccinology is going, the preparation of vaccines. So I'm sort of speaking on behalf of the National Council for Science Museums, and I do have a stake in it because the scientific part of it, I chaired that committee who put together this entire exhibition. So it's also my responsibility to make sure that many, many people actually come and visit that exhibition. It's really been put up very well. Take your time, go to Science City with at least 2 hours on hand. If the teachers can organize for schools or classes of schools or colleges to come and visit the vaccines exhibition, it'll be absolutely wonderful. Okay, so we'll get on with the first film, which is I think 7 or 8 minutes long. Talks about the Human Genome Project. Are you able to hear? Yes.
In 1989, a group of scientists was assembled to decode the human genome under the direction of Dr. James Watson.
One of Watson's big motivations here was to try to recruit the best and brightest from many different disciplines, biology, but also physics, chemistry, engineering, because all of those would have to get together if this was going to work.
Many of the biologists, and I love them dearly, of course, were dragged into it kicking or screaming in the beginning because they failed to see the tremendous value that such an enterprise would provide the field.
The talent turned out to be all over the world, so you have to basically make it possible for those bright minds to get engaged where they are. They're not going to all move to join this effort.
Genomics has fundamentally changed the way we do science. It brought the notion of team science and collaboration to biology.
Curious thing about collaboration, it's also intensely competitive. Each person wants to achieve something personal. They want to make their hit, on the problem, but then immediately they want to share it with the other, and the other learns immediately. And so you ratchet each other up.
From the beginning of the Genome Project, there was a fairly strong sense of enthusiasm in the public sector, the private, and academia.
We had lots of support from Republicans, partly because Newt Gingrich was interested in science. and I knew it. There's no partisan difference in everybody wanting to live as long as they can.
I think there's a lesson there. It isn't a big monolithic project. It was a big challenge, but it brought people together. And I think that's what we have to do in the future.
When the Human Genome Project was started, it's important to note the technology of sequencing was not developed yet. So it's, in a sense, a project that was launched with a wing and a prayer.
We couldn't sequence it at that time. The machines were nothing like fast enough. There were no machines even. It was sequencing was done by hand.
I thought we might have to do it brute force, and I was quietly prepared to use the old-fashioned technology just because it was so important to get it done.
There are a lot of things, that would make life easier that just hadn't been invented yet.
It would be a 100-year project like monks working to copy a book at best.
Bit by bit, we got into more and more large-scale sequencing. But the sequencing is enormously important because it provides the possibility of searching by computer.
When I showed up at Washington University one day with a computer terminal kind of under my arm, people looked at it and said, "What are you going to do with that?" This is a genetics department.
Genomes were supposed to be too complex to be able to reassemble them in the computer.
Without computers, we simply would be dead in the water.
Computers and genomics were made for each other. Genetic information is digital. It's like binary code. It just happens to use 4 digits instead of 2. It is amazingly accurate. And that's what biology gets from digital information.
The Human Genome Project contributed a cultural change about data sharing because the Genome Project was about sharing data almost immediately. And while it seems obvious now, it wasn't obvious then. That required significant effort.
I think I'm quite lucky as a scientist because I grew up always sharing data. The community around me, the genomics community, the worm and fly communities as well. It's just a default that you're going to share everything. You're going to share the code. You're going to share everything by publishing, but you're going to share it before publishing. You're going to share half-cooked ideas. And in fact, this is a wonderful thing. It's liberating.
It would have been virtually criminal to try to hoard discoveries that meant the difference in life and death in people.
This is information which we'll be using for centuries. The sooner we get it and everybody has access to it who wants it, the faster we can move in terms of understanding how life works and how disease occurs.
Patients invest a certain degree of fear about genetics. Who's going to find this out? How will it be used against me?
A lot of the ethical and social considerations that we're struggling with today really have historical roots in the early 1900s in the United States, where we had many states that had laws that allowed us to forcibly sterilize people and prevent them from having children based on whether we as a society felt that they were genetically fit.
So at the beginning of the Human Genome Project, we got it right of bringing in the social scientists, the ethicists, the lawyers, and we now have helped to provide a whole generation of scientists that are focusing on the ethical and social issues of genomics.
The sequencing of the human genome was a monumental achievement. Decades of work, thousands of scientists, billions of dollars, and we get this first roadmap for what makes up the human genome. It's an incredibly exciting moment. At the same time, it's just the beginning of what's really a much longer voyage.
And let's again be audacious, but let's not be unrealistic about what's possible.
I think the next 10 years are going to see some exciting developments in genomics, particularly those where we start to use genomic information as part of routine medical care. And that'll be applied to dealing with diseases like cancer, hypertension, diabetes, and Alzheimer's disease.
I dream of the day when every young mother brings a young girl or a young boy home from the hospital with a little gene card, and they say, this is the way you should prepare this child's health care. It's exhilarating.
So I hope that you enjoyed the film. I do want to make two announcements. One is an acknowledgement. You have seen Eric Green. You may or may not have followed his name. I got this film from Eric Green. Eric Green has been a friend for a very long time, an architect of the Human Genome Project, and he has visited the National Institute of Biomedical Genomics multiple times, was very instrumental and very supportive of the making of the new institute called National Institute of Biomedical Genomics. Not so new now. It started in 2009, 2010, and it's become more and more vibrant over a period of time. The second point that I want to make is that through the film, you have seen how this Human Genome Project has started, and there are multiple components to the project. One, of course, they mentioned that even the technologies did not exist, meaning that you had to start by doing it manually, and then machines came, and then there was a huge change in the way that you sequence human genomes, because essentially it's like a necklace, right, with beads on a string, and they were trying to read one bead at a time and then try and understand the entire string together. But then came this private institution called Celera Genomics, which was headed by J. Craig Venter, whom you have seen, and he completely changed the ball game. He said that I'm not going to do it end to end. I'm just going to break the necklace and do single small pieces and then put them together. And that's when the need for computers came, and they were already talking about Patrinos was mentioning that just the technologies wouldn't help. If we didn't have the computing equipment, we couldn't finish the Human Genome Project. So what I'm going to announce is that, and they have also talked about the ethical considerations of the human genome, the information that comes out of the human genome. So there are these multiple facets. And if schools are interested, if BITM is interested, I have not discussed this with members of the Calcutta Consortium, with the president of the Calcutta Consortium. What we might do is to arrange for a whole day meeting with some of us giving talks with illustrations, etc., on the various aspects of the Human Genome Project. And my talk, I've titled my talk as, what's all this excitement? There is indeed a lot of excitement, and I'm going to share some of this excitement with you. Some of you may already have heard this talk, or at least portions of this talk. So there is a lot of excitement, and if teachers from schools think that it is worthwhile for their students to know these various facets of this excitement, we'll be happy to come and do a whole day program in BITM. This is fairly central, so we could either do it here or we could do it in the National Council of Science Museums or in some other institution. Whatever suits you, we are offering our help to be able to expand on this theme. All right, without wasting more time, spending more time, let me go to my talk directly. So my talk is essentially a series of stories, and that's all. Do we have because I'm going to turn around, so do we have a collar microphone? Yes. Can I get a collar microphone? Because I'm going to turn around, and therefore I'm going to lose this microphone. It's better if I can get a collar microphone. All right. So, so this is what the title is all about. I'm going to talk about genomics. It all started with the Human Genome Project. Prior to that, it was essentially genetics that we were talking about, because we couldn't look at our entire, and I'll define the genome, we couldn't look at the entire, our genome in its entirety. So because we can now do genomics, there is a lot of excitement, and I'm just going to share some stories of excitement with you without getting into the nitty-gritty details of it, primarily because time will not permit me to get into the details. And anyway, like I have offered, that we can get into the details through a series of talks that we can do over an entire day, covering various aspects, various technical and other aspects of genomics. All right, so I'm going to show you a picture first, and let me see how many of you can actually identify this lady. Is there anyone who can identify this lady? Yeah, louder? Maybe you're too young to be able to identify this lady. She's a supermodel, as you can see from her figure, right? She's a supermodel. Her name is Nayanika Chatterjee. Now I can see some heads nodding, which essentially means that you have heard this, heard her name. And the reason why I'm showing her photo is because there was an article in the newspaper called The Telegraph, which essentially said that the secret behind Nayanika Chatterjee's fab figure, good genes and right diet. So essentially all of us, whatever we look like, however we behave, it's an interaction between genes and diet. At some point of time, and environment, diet is a part of the environment, at some point of time, many biologists felt that genes are not so important. It's the environment that's important, that makes us, what we are. But over a period of time, we have learned, we've seen such headlines as well, genes take charge, diets fall by the wayside. So genes became more and more prominent as we understood the genes better, the workings of the genes better, and how it interacts with the environment. And so over a period of time, environment became less important. Genes, we are studying genes, and genes actually are taking charge of various, our physical features, the way that we behave, etc. So this is another story that I'm going to share with you. This is, I was traveling, taking an international flight at some point of time, and you know that in the airlines there is this magazine, right? So the airline magazine had this picture. It's an advertisement of a cream, of cream that is called Genifique, and it's of course manufactured by a very famous company called Lancôme, and this company, everything from this that comes out of this company is very, very expensive. So this cream is also expensive. But what attracted my attention is that it says youth is in your genes. Reactivate it. Discover the skin you were born to have. So because we expose ourselves to various kinds of adverse environments, even if we are born to have good skin, the skin gets damaged, and this apparently repairs the damage. So that's what it said, and there is something that's written here which you can't read, which I can barely read from this distance, and it says that at the very origin of your skin's youth, your genes. So the youthfulness of skin comes from the activity of the genes or expressions of these genes. By the genes, by expressing themselves, they produce specific proteins that can be found on the skin's surface. So what they have done is to mimic what is normally produced by good genes, and they put it in a bottle, and they're asking you to buy the bottles to essentially rejuvenate your skin. This is what all of these cosmetic companies do. Some of them work, some of them don't, but it's interesting that these cosmetic companies, the manufacturers, are actually talking about genes and selling genes in order to sell their products. That's essentially what it is. So genes have become an exciting thing and have become an excitement. This is what attracted my attention. Triumph of genomic medicine. So even when we fall sick, the various kinds of drugs that we take are based on genomics, and that's the whole field that has been introduced, which is called genomic medicine. The New York Times says the decoded human genome, and we have heard about decoding of the human genome. This is a cosmic triumph for biology. So the way that New York Times said is that this first sequence of the human genome, which is essentially decoding the human genome, is a cosmic triumph for biology. It's like the cosmos. It's like capturing the cosmos, and that's the triumph of biology. Genetic study gives hope of cure for deadly diseases. So as through these examples, I hope that I've been able to convey to you that there is a smell of excitement in the air. And that's the excitement of genomics, and therefore we should learn genomics. And again, what I'm going to do is tell you a series of examples that are not so advertisement-like, but more scientific. So essentially, that's what I'm going to do through the rest of my talk. You have seen these three people. This is Collins, Francis Collins, who was head of the National Institutes of Health when the Human Genome Project started. The National Institute of Health was one of the major players of the Human Genome Project in decoding the human genome. This is Craig Venter, of whom I mentioned. He was an employee of the National Institutes of Health. And he completely disagreed with the leadership of the National Institute of Health because they were trying to read first base, second base, third base, fourth base, fifth base. He said, no, I'm not going to do it that way. This is going to, I'll die before the first gene gets decoded. So I want to see the genome decoded before I die. So he came up with this alternative. He said, I'm going to break this necklace, chop this necklace randomly into small parts, and I'm going to sequence each of those parts and then reassemble them together. Nobody believed that you could actually reassemble. So he founded a company called Celera Genomics. They came up with these various kinds of software packages and very fast machines that could actually reassemble these broken pieces and put them together to actually identify the entire string, the beads on the entire string. And this is how eventually the Human Genome Project was done. So this is our genome, and we, as each of us know, that this is a double-stranded molecule. Only one of them is essential; the other is completely redundant because A always pairs with T and C always pairs with G, and therefore, if you know the sequence of one strand, you automatically know the sequence of the other strand. And there are only 4 alphabets in this sentence that we are trying to read. So that's how the sequence looks like. It's just a bunch of A, T, Gs, and Cs. We need to figure out where these A, T, Gs, and Cs are. And there are the strand is about 3 times 10 to the power of 9, which is 3 billion of these little alphabets. How are these 3 billion alphabets stacked? They are stacked in 23 pairs of chromosomes. So each one of these chromosomes will have a portion of our genome, and the DNA that's embedded in the genome, the entire code that's embedded in these 23 pairs of chromosomes is essentially what our genome is, and that's 3 billion base pairs. How long did it take? Even after Craig Venter came in with his new philosophy of breaking the strand and sequencing small bits of the strand, it took 13 years to complete the first sequence of the human genome. 13 years, it took $1 per alphabet, $1 per nucleotide, 3 billion base pairs, 3 billion US dollars is what it took to sequence the first strand, first human genome, and it took 13 years to do so. Now let me tell you, come to Kalyani, come to the National Institute of Biomedical Genomics, reach out to Dr. Arindam Moitro. Give him a small sample of your blood, 1 milliliter of your blood. He will tell you, come back after a week with about 2 lakh rupees in your pocket, I will give you your sequence. 1 week, 3 billion US dollars, 2 lakh rupees, maybe even lesser than that. He may have reduced the cost now. 2 lakh rupees, 1 week. That's how rapidly we can do this. And there have been huge technological advances. 13 years to 1 week, $3 billion to 2 lakh rupees. Yeah, this is how the scales have fallen. Scales of time and scales of cost has fallen. This is because of improvements in technologies of sequencing. I do not have the time now to talk about technologies of sequencing, but one of these days, if you do a full day workshop here, we would be able to describe to you how these sequencing technologies have actually improved. So this is what it is. So genes are decoded to produce proteins. Proteins are the ultimate things that we require, right, for our physiology, for digestion of food, for how we behave, how we talk, and so on and so forth. And these codes control the genes. The decoding of the human genome, that's a code. That specific code is decoded, and they control the genes. And sequences which we still don't understand. So genes we still don't know the exact number of genes in the genome, in the 3 billion alphabet. So what we do know is that the gene number varies between maybe 15 and 20,000. The remaining part, the 15 and 20,000, comprise between 1 and 2% of the entire genome. The remaining 99 to 98% of the genome, we understand some of the function. They mostly regulate these genes, but we also don't know large chunks of the function that they may be doing, and therefore sequencing these human genomes will actually enable us to understand, we hope, to understand what these 97, 98, 99% of the genome that we don't understand yet actually do. They just can't be. At some point of time, we felt that it was junk DNA. Many people believe that it just can't be junk. There must be some function to it which we don't understand. And so all of you have a huge field day in order to be able to identify the function of these portions of the genome that we still do not understand. The other thing that I want to mention is that after the first genome was decoded, after the first human genome sequence was published, people realized that we behave differently, we talk differently, we look different. All of this are because our bodies are producing, our genes are producing different kinds of proteins. So we just can't, our genomes can't be exactly the same. There will be a lot of variation between my genome and your genome. So there is a lot of variation. So the next major international project that came about was to understand the extent, the nature and extent of variation in genome sequences across individuals, because that would probably inform us as to why we behave differently. Let's say that we find a bomb burst. Some of us run away from the bomb. Some of us will stand there and stare. When is the next bomb coming? So why do we behave differently, right? So that probably is in our genes, and therefore we need to understand the variation, and that's the reason why we need to understand the variation. And the next major project that was undertaken is called the Human Genome Variation Project, and this is the Human Genome Variation. That became the breakthrough of the year. Do I have the I can't read the date. So anyway, this was done after the Human Genome Project, and eventually we now have, like Arinda mentioned, that we began in this city to understand the variation among genomic variation among people in different parts of India. It has been taken to a much greater height by people like Professor Onulabo Basu, who has just sequenced 10,000 human genomes of 10,000 individuals sampled from all over India. So we now have a database to understand, why different people in different parts of India look different, and so on and so forth. So that data have just been collected, and it's just being analyzed, and hopefully will provide us with a lot of information about the different population groups of India. But we already do have a lot of information about different population groups of the world, how their genomes vary. So the variation is like, both strands are not necessary. If we look at G there, we have A here. So these are two individuals, the same position, that's G there, and this is A here. They probably, that triplets produce amino acids. These two triplets, GGC and GAC, probably produce two different amino acids, and therefore their proteins may actually be quite different. So this is the kind of variation that we want to study, and this is the variation that is being studied, and more data are being generated. And there is now another move to start a Human Genome Project 2, which will sequence very, very large numbers, or maybe million individuals in the next phase. All right, so as I said that I'm introducing people. How many of you know this little girl? Nobody has met her. Do you know her name? No? I'm surprised. Her name is Tina. You've met her, right? So I'm going to tell you about her story. She has a life. I'm going to tell you about her story, and this story has some morals about what we should be doing now that we have genome data on large numbers of individuals. So let's see. This is Tina's story in 2020. Tina has a careful family history taken at age 25, learns of uncles with early heart disease. So she has some uncles with early heart disease. She consults epidemiological literature. Epidemiological literature looks at, what kind of, what is the prevalence of a disease, what fraction of a population suffer from a specific disease such as heart disease, etc., etc. So she consults that kind of literature and finds out that 3 in every 50,000 individuals about the age of 25 in her district suffers from early heart disease. Her age is 25. 3 in every 50,000 individuals. Early heart disease can be prevented by regular exercise and fat-free diet. Some medicines called statins are available to reduce the risk of heart disease accumulating from poor diet. And to her dismay, she picks up a recent paper in the New England Journal of Medicine. The New England Journal of Medicine is the oldest medical journal in the world. She picks up a paper, a publication in the New England Journal of Medicine, in which data are presented to show that not everyone can benefit from taking statins. So you take statins to reduce the risk of heart disease accumulating from poor diet, but even if you take statins, you may not actually benefit because not everyone can benefit. And this has been shown in this particular scientific study that was published in the New England Journal of Medicine. By the way, you've met Francis Collins. So this is a story that, over multiple cups of coffee in a meeting in China that we concocted this story. So this is not my authorship. Francis Collins is also an author of this story. Tina's story continues. She consults her physician, who has made an effort to stay informed about genomics of complex common diseases. Heart disease is a complex disease. It's not due to a single gene, or it's not due to your eating butter every day. There are other kinds of interactions that go on. So that's a complex disease, and it's very common in the population. The physician suggests complete genome sequencing for 60,000 rupees. So you're being a little futuristic. The costs are coming down. So right now, Arindam can do at 1.52 lakh rupees. So a day will come, maybe next year he'll be able to do it at 60,000 rupees. She's found to have 3 gene variants, and I've told you what those gene variants are. Variants are, positions on the genome at which not everybody has exactly the same nucleotide or the same base or the same alphabet. She's found to have 3 gene variants that have been shown conclusively in well-validated studies. The validation is an important component of every scientific study. You can make a discovery, but that discovery oftentimes is false. So you have to replicate this discovery in a new set of individuals or a new set of patients and see that the same discovery actually validates in a new set of individuals. So validation is an important component of scientific studies. So she found out that these 3 gene variants have recently been conclusively shown to increase the risk of early heart attack 4-fold. Increasing the risk of early heart attack 4-fold is really a danger to the individuals who may have these gene variants. She and her physician design a proper program of prevention based on diet, exercise, and medication precisely targeted to her genetic condition. Today, everybody takes the same drug, but the same drug doesn't act on everybody. So we need to think of the day that's coming where medication will be targeted to my genetic situation. She has 3 gene variants, and therefore the medication, so this is a little futuristic, the medication that she would take would be precisely targeted to her genetic condition. You can't change the variants in the genes, right? We still don't have the ability to change the gene variants. So what we need to do is to take the medication that's appropriate for that genetic condition, and we must take diet because this is heart attack. We must take probably fat-free diet and do exercise to burn the calories and so on and so forth. So they design a program of prevention. Her story continues. Tina does well until the age of 75. She develops left arm pain that she assumes is due to gardening. Tina loved gardening. She would grow flowers. She would spend many hours a day doing gardening. She assumes that the left arm pain that she has is due to gardening, but her physician knows her higher risk and diagnoses that she's undergoing an acute myocardial infarction. Myocardial infarction is a heart attack. So she's probably undergoing a heart attack. She thinks that her left arm pain is because of gardening. Referring to her genome sequence, the physician chooses the drugs that will work the best to treat her. Today we don't yet, but this is kind of futuristic, and therefore the physician chooses the drugs that will that will work the best to treat her. And those drugs are given. she survives and is alive and well until into the 22nd century. So this is all fantastic. This is all wonderful. This is if we do our genomics well, if our physicians understand genomics, if our physicians understand what kind of gene variants, what kind of drugs work under what kind of genetic conditions, then our lives will become much better. But this will take some time, so it's kind of futuristic. Tina's story gone wrong. There's a very likely chance that unless we inform, unless we do research on genomics, unless our clinicians adapt themselves and learn genomics, then we are not going to, unless drugs are discovered with respect to our specific genetic condition, we are going to be in deep trouble. And Tina's story gone wrong. Tina never learns about her family history. Learning family history is the first component that's very important. Educational efforts for the public and physicians were never mounted, so you need to have these kinds of programs, right, like we are doing, such that the public and physicians, you're much better educated than the public, but public and physicians were never mounted, and Tina's physician thought genetics was irrelevant to practice, to medical practice, which we now know is completely wrong. Tina loves ice cream, chocolates, and mutton. She soon gains weight and develops high blood pressure. While tests to predict which drug would be most effective for Tina have been proposed, they have never been validated and are not reimbursed by Mediclaim. So you need somebody to pay for the drugs, and insurance, she pays insurance, but the insurance company doesn't pay for these drugs. Not all drugs are covered by the insurance companies. And so some of these drugs are not covered by the insurance companies. These, because they have not been validated, the insurance companies will not pay you, will not reimburse you for those kinds of drugs, and therefore this is a sad story. Tina's hypertension is treated with a drug that causes hypersensitivity reaction, so she stops treatment. As soon as she takes this hypertensive medication that reduces her blood pressure level, she starts itching, she starts scratching her whole body. The body becomes red. So that's terrible for her. She doesn't like it. Her increased blood pressure she cannot feel, but she can certainly feel the redness on the skin and the scratching, so she stops treatment. After 10 years of uncontrolled, because she stopped treatment, the hypertension, the blood pressure levels are uncontrolled, Tina develops left arm pain at the age of 50. Unaware of her high risk, her physician assumes that this is musculoskeletal and prescribes rest. The physician is also not well informed about these various developments in genetics. Tina returns to the emergency room of the hospital in a few hours. A few hours later, she was at home for a few hours and goes to the emergency room of the hospital in cardiogenic shock. The absence of a genome sequence information prevents immediate optimum choice of therapy. So she's given a generic drug instead of a drug that will suit her more precisely because genomic information is not available. Tina dies in the emergency room. So here is one part of the story about Tina that I proposed or narrated, where knowing genomic information, we could actually save Tina. She lived into the 22nd century. Here, Tina dies in the emergency room primarily because neither she nor her physician was aware of genomics, and people who make drugs or discover drugs, they also didn't actually discover or get into the discovery of drugs based on genomic sequences. That's what happens. If we don't learn genetics, genomics, and see how it caters to our body, caters to our physiology, this is what we are going to face with. We don't want to face that, so we need to learn genomics. There is a lot of excitement about genomics. What's the charge to all of us? The charge to all of us is save Tina. It's not just one Tina, Tinas of the world. So this is what we need to do, and that's the charge for all of us. So what's the relevance of Tina's story here? We should learn genetics and biotechnology. Of course, as I told you, that much of genomics today is dependent on technology. So we should all learn genomics and biotechnology to execute the charge that's given to all of us, which is to save Tina. All right, so I'm trying to enthuse you to learn genetics and learn these various technologies associated with genetics so that there will be no more Tinas who will die in the emergency room. A few more stories and I'll stop. This is Mary, the Queen of Scots, Queen Victoria, and Stephen Hawking. Very different times of very different times during our during humankind. The question is, what do they have in common? And I'll give you the answer to the question. What they have in common are genetic variants that have caused diseases. So this is, I told you about taking family history. This is how we take family history. The horizontal lines are individuals who belong to the same generation. The vertical lines are individuals who belong to different generations. The ones that are marked red are ones who have the disease, and the ones that are hollow, not marked, not filled, are ones who do not have the disease. As you can see that there are many individuals in this particular family who have the disease. And where is Queen Victoria, Queen of Scots? This is Mary, the Queen of Scots, and she has this particular disease. And what is this particular disease? This particular disease is called porphyria, where the outcome is an acute abdominal pain, nervous and psychiatric symptoms. These develop over a period of time. You start with bouts of acute abdominal pain, then you start feeling nervous, you start shaking, and then you have psychiatric problems. You start going nuts. The second lady, which is Queen Victoria, and again, as you can see that this is her family history, many individuals affected, she herself is affected. This is a little different because as you could see in the previous graph, there is no individual who is half filled. Every individual, circles are females, squares are males. Every individual is fully filled, but here Queen Victoria herself is half filled. The reason being that Queen Victoria, that this particular gene called hemophilia, where if we get a cut, most of us, if we get a cut, after it'll bleed, and after a while it'll actually coagulate, and the bleeding will automatically stop. But if you have a genetic variant that causes this disease called hemophilia, you essentially bleed to death. The coagulation doesn't happen and you bleed to death. This is I'll come back to this, why it's half filled. The third individual, Stephen Hawking, he has a disease called amyotrophic lateral sclerosis, or ALS. Go back to Mary. She had this particular disease called porphyria. We now know porphyria is determined by a variant in a gene called ERO-SSI and IIIS, and this gene is on chromosome 10. We have 23 pairs of chromosomes. This is on the 10th chromosomal pair. Each chromosome has a centromere and two arms. Usually the centromere is not right in the center. It is shifted away from the center sometimes. On chromosome 10, so there are the two arms have a long arm and a short arm. The long arm is usually denoted as Q, and the short arm is usually denoted as P. And this is on the long arm, and this 24 or 24 as we call it, the long arm is banded, and this is a specific band on the long arm of chromosome 10 that's called 24. So this particular gene is on the location of this gene is on the long arm of chromosome 10, and on the band 24. So we now know the location of that gene also, and a variation in this gene causes the disease porphyria, which is acute abdominal pain, nervousness, and eventually psychiatric problems. This is a gene that's on the X chromosome. So if females have two X chromosomes, males have one X chromosome. And this particular gene behaves as a dominant gene, and that's the reason why it was half marked, because she has only one copy that has a specific variant, and this has to do with blood coagulation, as I mentioned. Factor VIII gene is a blood coagulation gene. It's located on the X chromosome, long arm of X chromosome, and right there this is the end of that long arm. This is the long arm, this is short arm. Right there in the end of the long arm of this chromosome called the telomere. So this is a telomeric gene, factor VIII, and there is a variant in that gene because of which Queen Victoria was going to bleed to death if she got a deep cut in her body. The third individual, amyotrophic lateral sclerosis, Stephen Hawking, this is a missense mutation in a gene called superoxide dismutase that's responsible not for all of these cases, but only explains about 20% of the cases. The reason why I'm putting up this particular slide is because the previous two cases were because of a single gene, and that's why anybody who got a variant in that gene would get that disease. But here is a disease where only 20% of the individuals would get the disease if they had a mutation or if they had a variation in that gene. The remaining 80% were due to other kinds of genes, but for Stephen Hawking, it was because of this particular gene. And what it does, we now know the effect of this particular gene, the function of this gene, and what happens if there is a mutation in this gene. All of that has been worked out. So essentially, by looking at diseases, by collecting carefully family history, by doing genomic work, you can map genes on specific chromosomes and identify what kind of variation leads to disease, and that's extremely important. And the next point that I want to make is that even if you have no variants in that gene, it is not true that you may not get that disease, because it's only 20% of these cases that's because of this variation in this particular gene. So essentially, these are unpredictable, and you need to make them predictable by studying genomics, and that's what we do. We try to understand what these genes do, and understanding actually enhances predictability. So many genetic disorders don't express themselves at birth, they express themselves later in life. But if you can do a genomic sequencing at birth, then we know that which particular diseases this individual is predisposed to, and then we can predict which diseases this individual is predisposed to, and we might take appropriate precautions right from birth or childhood, such that we can at least delay the onset of that disorder, even if we cannot completely prevent the disorder. So there are now medications that have become available. There is a recombinant factor VIII. Recombinant is a technology. Again, I said that I emphasize that not only genomics, but also biotechnology is something to learn. This is recombinant DNA technology is like a biotechnological phenomenon. And because of that, there are now factor VIII powders that are available, which are used to treat these individuals who after getting a cut actually bleed to death. So understanding, and because this was understood, why that coagulation does not happen, this understanding enhances treatment as well, not just predictability, but also treatment in some cases. Lots of people know her, right? How wonderful. Many people know her. When I saw her picture, I had to read the article to figure out who this lady was. But do you know why I'm showing her picture? Does anybody know why I'm showing her picture? I'm going to tell you her story, but do you know why I'm showing her picture? All right, I think some of you know, but you're not sure. So that's why I hear mumbling, but not a clear answer to my question. Of course, you know who this lady is, right? Angelina Jolie. And the reason why I'm showing you her picture is because she also took family history and figured out that her mother, Marcellina Bertrand, that was her name, she died from ovarian cancer at a relatively young age of 56. One doesn't die of ovarian cancer at such a young age, but she did. And then after when she was being treated, they figured out that she had a faulty gene, which actually she had passed on to Angelina. So Angelina also got herself tested for that faulty gene and figured out that her mother actually had passed on that faulty gene, meaning the gene with the variant, with the faulty variant that predisposes you to that disease, to Angelina. So she had a faulty gene which she passed on to Angelina, and therefore Angelina didn't get ovarian cancer yet. So and this gene that predisposes one to ovarian cancer also predisposes to breast cancer. So Angelina thought that I might either get breast cancer or ovarian cancer. I must protect myself. So what did she do? The first thing she did is that these are organs of the body, right? Breast and ovaries. So let me get rid of them such that they don't become affected. That's what she did. She actually removed her breast. She underwent radical mastectomy, got herself surgically got her breasts removed. And once, so these are two genes, BRCA1 and BRCA2, that predispose you to predisposes a woman to breast cancer, and increases the risk by about 5-fold or 6-fold. And there's another gene which is less harmful, meaning less impactful, on disease, so which increases the risk by 45%. So this is breast cancer. This also increases the risk to ovarian cancer, and BRCA2 also increases the risk to ovarian cancer. So essentially the same alterations in these two genes, BRCA1 and BRCA2, predisposes a woman to both breast cancer and ovarian cancer. These are technically called tumor suppressors, but let me not get into the technicalities. So once she got her breast removed, lots of ladies in the United States decided that we must get ourselves tested, and if we do turn out positive, then we'll think whether we want to get our breast removed. So testing increased 40% as soon as the general public learned that Angelina Jolie had that faulty gene and she had undergone radical mastectomy, which is the technical term for surgical removal of breasts. Not only did she remove her breast, a year later, she actually removed her ovaries and fallopian tubes. Is this the way to understand genes and to treat yourself, get rid of? If she had a gene that predisposed to lung cancer, what would she do? She would remove her lungs? Wouldn't be possible, right? So this is not the way that you use genomic information. This is not the way to use genomic information. You need to of course, understand your genome, take preventive action, but preventive action doesn't mean that you need to remove organs of the body. I'm going to talk about children. I'm going to take another 10 minutes. Is that good? Is that okay? So I'm going to talk about children. So I've talked about, well, let me talk about children. So this is a disease it's childhood leukemia. So this childhood leukemia, the percent of survival children who survive from this kind of leukemia, it's a specific form of leukemia. I don't want to get into the technicalities. It's leukemia of a specific kind that the percent of survival children has increased from by over 20-fold in 50 years. In 1960, only 4 of 100 children would survive from this leukemia. In 2014, 92 of those of the 100 children who suffer from leukemia actually survive. So the survival probability has hugely increased. How did this happen? This happened because we understood the nature of genomic alteration that takes place. In this case, it's a gross genomic alteration, and I'll explain that in a moment. It's not like a single nucleotide change. It's not like a single base substitution that's happening in a gene. It's more gross than that. It's much more impactful. This is called a fluorescent in situ hybridization, again, a technology that allows us to understand these various kinds of gross chromosomal anomalies that take place. I'm not going to explain this. No, let me just skip this. But again, I do want to point out this particular book. The lady who actually found out this, the entire background to this disease, this form of childhood leukemia that actually enabled survival from, increase in survival by about 20-fold, that's all been enshrined in this book called the Philadelphia chromosome. And I'll mention about the Philadelphia chromosome in a minute. And this actually has the history of discovery of the biological defect that underlies this particular disease and what actions were taken subsequent to understanding the biological defect that leads to this disease. So this is a normal chromosome 9, this is a normal chromosome 22, there are 23 pairs of chromosomes. This chromosome 9 has a gene called ABL, and this chromosome 22 has a gene called BCR. In most individuals, these two genes would produce certain kinds of proteins, etc., that are helpful. Sometimes a gross biological change takes place where this part of the chromosome, of chromosome 9, and goes and attaches itself to chromosome 22, and this part of chromosome 22 goes and attaches itself to chromosome 21. How this happens, etc., it's for you to learn in biology, but this happens. Not very frequent, rarely, but it does happen. And then what we get is what's called the Philadelphia chromosome. So this chromosome 9 gets elongated, and this chromosome chromosome 22 gets shortened. But what happens as a result of this is called translocation. What happens as a translocation of this part of the chromosome to this part of the chromosome? What happens as a result of translocation is that these two genes that were on two separate chromosomes were producing two separate proteins. They come together and they start producing what's called a hybrid protein, and this hybrid protein is dangerous. It actually produces this disease called leukemia. So this was understood And once this was understood and the functions of these two genes independently were known, once the function of this hybrid gene got to be known, then people came and drug companies came and they found a gene, they found a drug that actually stops the protein is produced, but then the action of the protein, they would use the drug in order to stop the action of the protein. And I'm not going to describe to you all of this. This drug is called imatinib. It was one of the first drugs that was biotechnologically discovered after the main discovery, the biological discovery was made. So this drug was able to save lives of children, and it used genomic information, it used biological information of this translocation. And you have seen how many lives of children are being saved. The increase in lives of children, lives of children saved increased 20-fold in 50 years. So that's a huge change, and this was enabled by the biological information and using the biological information, finding of a new drug called imatinib. So this is the lady, her name is Janet Rowley. She's the one who found this translocation. This is called the Philadelphia chromosome. The translocated chromosome that produces this hybrid protein that chromosome is called the Philadelphia chromosome because she worked in the Philadelphia Medical School of Pennsylvania in Philadelphia, and that's the reason. That's where she discovered it, and that's why it's called the Philadelphia chromosome. It was on September 22nd is when she announced her discovery, and this is celebrated every year as the World CML Day. This L stands for leukemia. Don't worry about what CM stands for. It's for that specific kind of leukemia. Not all leukemias are because of 922 translocation. Not all leukemias are because of that. It's only one form of leukemia that's because of this 922 translocation. She passed away in December 23, and this is Barack Obama who actually presented her with a national gold medal, with National Medal of Science, which she won from because of her discovery and because of the fact that her discovery led to saving of lots of lives. I'll turn to children now, turn to women now. Worldwide, 1.6 million new cases of breast cancer are diagnosed each year, leading to over 500,000 deaths. And fortunately, these 1.6 million people do not take the path that Angelina Jolie had taken. 40 years ago, the 5-year survival rate, once you were detected with leukemia, with breast cancer, 40 years ago, the 5-year survival, the probability, the chance that you would survive for 5 more years was only 50%. Today, that figure is 80%. If you get diagnosed today with breast cancer, the chance that you will live for another 5 years increased from 80% to from 50% to 80% in about 40 years. How did that happen? Because now we know that in about 20% of breast cancers, a specific protein that's known as HER2 is overexpressed. So that it makes too much of a protein known as HER2. And once in a woman, this particular HER2 protein is overexpressed or overmade, then these cancers tend to be aggressive and fast growing. And therefore, one needed to sort of temper down the extent of production of this particular protein. This is on chromosome number 17. That's the location of this HER2 gene. And first of all, it was located. What the change was that led to overproduction of the protein was understood, and eventually, I'm not going to get into that. So eventually a new drug was identified, which is called Herceptin, that dramatically reduces the risk of breast cancer coming back. So because it dramatically reduces the risk, so you survive for a longer period of time. Also, many of these drugs have side effects, but Herceptin has far fewer side effects. For example, you start to vomit once you take these kinds of drugs, and Herceptin was. I'm going to skip this. So, based on the biological information that one found that this Herceptin or HER2 gene, overexpression of the HER2 gene led to this form of breast cancer, a drug was discovered, and that drug is saving lives. So it's saving lives of women by tailoring drugs more precisely using information on their genomes. So we saved lives of children by understanding translocations. We are now able to save lives of women by understanding the kind of variation, that genomic variation that takes place in this HER2 gene. Why am I going back? The last part, another 5 minutes, saving lives of men, and I tell you two stories. This is a person who came to a doctor with this kind of it's a scanned image. It's called a PET scan, scanned image, and each of these black dots is a tumor. So this person has tumors all over the body. Came to the doctor looked at this image and said that I'm going to give you this drug and I will watch you. So the drug was given. After a few weeks, after about 6 weeks, all the tumors have vanished. They vanished into thin air. The drug was acted so well that you can see that all of these dots are gone from this. This is an image that was taken 6 weeks later. Was it 6 weeks or 6 months? I can't remember. This is a true story that was published in that journal that I mentioned, New England Journal of Medicine. It's from the pages of that book. Another person with roughly the same kind of an image came to the doctor a few years later, and the doctor was absolutely certain that he would be able to cure this person by giving that drug at the same dosage level for the same duration of time. The person would go to this particular state. Unfortunately, this is what happened. Not one tumor regressed. There was not one tumor vanished. This person remained at exactly the same condition, even though this person got exactly the same drug that the previous person had gotten. So the question is, why is there a smiley here and a sad face here? We'll try and understand that. The second story involves me, or my personal association with this story. This is a conversation that I overheard on the corridor of a particular hospital, and this is between a doctor and the wife of a patient, and the patient was a teacher of mine. I studied at the Indian Statistical Institute, was a teacher of mine at that institute, and he was a chain smoker, had lung cancer. Don't worry about what this form of lung cancer is. This is that last LC is lung cancer, had lung cancer. So the doctor, this is what I'm overhearing. The doctor and the wife of the patient are walking in front of me. I'm towing them from behind because I referred the patient's family to the doctor. I know the doctor, I know the doctor, so I referred. So the doctor says, well, the patient has lung cancer. I need to put him on chemotherapy. As soon as the wife hears about chemotherapy, all her hair goes straight up. Chemotherapy is something dreaded, right? And also, she lives quite far away from the hospital. She has to undertake maybe 3 or 4 hours of drive every day to get to the hospital and so on. So she asks a natural question, can the chemotherapy be done at home? The doctor says no, then pauses for a while and says, let's check out his EGFR mutation status. EGFR is the epidermal growth factor receptor. That's a gene. So the doctor was well informed about genomics and says that let me check out his EGFR mutation status. If there are variations from normal, then what we'll do is we'll take a different course. If it turns out that he has these variations in the EGFR gene, then you can take him home and administer chemotherapy orally. So again, this is a conversation that I overheard, and this is extremely nice that the doctor was informed about genomics and said that if it turns out that he has variations in this particular gene, you can take him home and administer chemotherapy orally. Oral chemotherapeutic agents have been found when patients have lung cancer patients have mutations in this particular gene. I'm going back. So essentially, he had mutations in EGFR, and he was able to be taken home, administered chemotherapy. I was going to tell you a little bit of biology of EGFR, but I'm going to skip that. So he lived for about 4 years, leading a near normal life. So usually lung cancer patients don't live for so long, but because he was given this oral chemotherapeutic drug, which was specific to those specific variations in the EGFR gene, he was able to live for 4 more years. Come back, and this is the last 30 seconds of my talk. Essentially, I talked about these two people and questioned myself, why is there a smiley here and a sad face here? The smiley here and the sad face here is because there are mutations in a gene called BRAF that's found in malignant melanoma primarily, but also in other kinds of cancer, including the kind of cancer that this person had. It's found in 70% of melanoma. Melanoma is skin cancer, 30% of thyroid cancer, 10% of colorectal cancer. What this gene does, you may know, those of you who study biology, that there is a lot of cargo. This is a cell, this is the cell wall. There's a lot of cargo, molecules, biological molecules that come from outside of the cell and are transported to inside of the cell when the cell requires that particular kind of molecules. And that there is a policeman who is standing here. The policeman's name is BRAF, and this particular policeman opens the door for those molecules to come in, and when the sufficient number of molecules that have come into the cell, the policeman will close the door. So that's the work of this particular policeman called BRAF, which is really a gene. Now, when this policeman is actually lost his hand, or his hands became paralyzed because of certain mutations, then the policeman would be able to open the door but perhaps not close it, because closing takes a lot of effort. So then the policeman will not be able to close it. So what usually happens, so this is what happens, it's a directed entry of these various kinds of molecules, and the door will close. It allows the cell to grow, and when the cell does not need to grow anymore, the gate will close and no more of that cargo will come in. But when the policeman is injured or something, cannot close the door and open the door, so essentially when there is mutation, then the cargo will just keep coming into the cell, and the cell will never stop growing. It will grow and grow and grow because the policeman is unable to close the door. So this is what happens when there is a mutation in BRAF, and that's a cancer cell. A hallmark of cancer cell is uncontrolled cellular growth, and the uncontrolled cellular growth in these forms of cancer are because of mutations in this gene called BRAF. So now we have a drug that where if the policeman is unable to close the gate, then the drug will close the gate. So essentially there is a drug that acts specifically against these mutations in BRAF. And you can now I don't even have to tell you the story. You can imagine that both of these patients had that form of cancer. One of the patients had a BRAF mutation. Not every patient will have BRAF mutation. This patient did not have BRAF mutation. So the drug worked very well on this individual because the drug works against mutated BRAF. But the drug does not work against those patients where the disease is because of mutation in some other gene and not because of mutation in BRAF. Then when there is no mutation in BRAF, the drug will not work. So that particular drug did not work in this patient, and that patient remained in exactly the same condition, even though the patient took that drug for as many at that same dosage for as many weeks as this patient had taken. This patient got cured primarily because this patient had that particular mutation, and this mutation is this drug is against that particular mutation. So genetics and genomics is touching our lives every day. And I've talked about children, I've talked about women, I've talked about men. I've given you a certain number of examples, didn't go deep into the biology. Well, I've given you some idea about biology and genomics, but I hope that you are as excited as I am, or at least more excited than I am, that genetics and genomics is touching our lives every day. And that's the excitement, and that's why I titled my talk as Genomics, what's all this excitement? And I've shared some stories about with you about the various kinds of excitement that's happening because of our knowledge of genomics. Thank you very much. So, for another 3 minutes, we'll see another film. And that's narrated by Siddhartha Mukherjee, whom many of you may have heard of, and I will show you that film. This is about the future of genomics.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
This was narrated by Siddhartha Mukherjee, a Pulitzer Prize winner. He's written a fantastic book. He's written several. First book that he had written was had won him a Pulitzer Prize. Some of you may have read that book. If you did not, look him up on the internet. Siddhartha Mukherjee is his name. He works in the US, but he's a doctor, he's a clinician, he treats patients. In addition, he's kept himself abreast of genomics and has actually reached out to the public to make the general public, genomics known to the general public. So he's a wonderful writer as well. I will stop here, and I don't know what Aditi has in store for us, but again, the point is that, like I said in the beginning, we are willing to come back and discuss more scientific details with you. Students make demand to your teachers, and teachers make demand on BITM, and we are there. BITM will reach out to us, and we can again hold maybe a whole day meeting, touching upon these various aspects that were shown in the first film that I showed, including the ethical aspects of doing genomics. Thank you very much. Thank you for your participation. Oito.